Prehistoric Marine Ecosystems by Molly Miller, Alexa Harris, Emily Allworth, and Jonathan Belfer. Researchers used a near-infrared femtolaser ablation multicollector ICP-MS technique, but we'll call it NIR-FS-LA-MC-ICP-MS system. They also used a conventional solution method to measure the isotopic iron ratio of marine sedimentary minerals. The study shows the microscale iron isotopic ratio of individual pyrite grains over a geological time period in shallow marine stromatolitic carbonates. Pyrite used in this experiment is also known as iron pyrite or fool's gold. The two main iron isotopes found in this experiment are iron 56 and iron 57, with iron 56 being the most common iron isotope. Shallow seas in the Precambrian era have been linked with material cycle and photosynthesis-based ecosystem. In order to test this, a section of pyrite was polished off and loaded into an aluminum-based sample cell. These findings include a range of iron and iron 2+ ions. The positive values of iron support partial oxidation. This was estimated by the iron 2+ aqueous steady state model, which implies that oxygen may have not accumulated in the atmosphere. These findings can help reconstruct the redox of shallow seas. 3 pyrite examples were tested: JMC pyrite, which was the standard, Ueno pyrite, and a pyrite in black shale. The Ueno pyrite samples agreed with each other in both the tests. The pyrite from the black shale varied at about plus or minus 0.042%. This graph shows the relationship between iron 56 and iron 57 ratios. The straight gray line is the theoretical mass-dependent fraction line. This data is consistent with non-mass-independent isotope effects. Results. The iron isotopic ratio of pyrite at 2.7 billion years shows that half of the iron 2+ aqueous influx was oxidized in the sea at the time. This means that oxygen produced by cyanobacteria would have been consumed by the oxidation of the iron 2 aqueous influx, and that oxygen probably didn't accumulate in the atmosphere. Pyrite, within 2.7 billion years, shallow marine stromatolitic carbonate from the Fortes group shows a unique bimetal iron isotopic distribution with highly positive and negative iron 56 values. Iron isotopic ratio of pyrite in the 2.6 billion Campbell Red subgroup is similar to that of the contemporary shallow sea aqueous iron 2+, which was estimated from the iron 56 value of shallow marine carbonate in the Campbell Red subgroup. It is reasonable to consider that the main iron source of 2.6 billion pyrite was seawater from iron 2+ aqueous. The iron isotopic ratio of pyrite is similar to that of iron 2+ aqueous due to rapid isotopic equilibrium. These results demonstrate that the grain-scale iron isotopic distribution of pyrite could be a useful index for reconstructing the redox state of the past shallow sea, as well as distinguishing biological and abiological iron isotopic signatures. This paper focuses on the change in ecospace utilization from the mid-Paleozoic time period until the late Cenozoic time period. These scientists followed the 3 characteristics to determine the ecospace utilization: vertical tiering, motility level, and feeding mechanism. By theoretical ecospace, the paleontologists mean that they are looking at all possible combinations of ecological patterns, not just those that are documented in the fossil record. How have modes of life changed over time? This graph is what the paper is based on. Each box represents a mode of life, which is a unique combination of tiering, motility, and feeding categories. The main goal of this study was to determine how they change over time. Tiering refers to the position of an adult organism relative to the sediment and water interface. It shows where an animal lives and seeks resources. The second factor they tested is motility. It is the ability of an organism to move under its own power. Attachment to a substrate is part of motility because it is a way for an organism to control its own movement. The third factor is the feeding mechanism, normally determined by observing an animal's morphology. This image reveals all the changes from the mid-Paleozoic on the bottom to the late Cenozoic seen on the top. This is another graph that perfectly shows the changes in tiering, motility, and feeding between the mid-Paleozoic and late Cenozoic. In the next slide, you will see the modes of life that the paleontologists found to be most dynamic. To compile this list, Bush, Bambach, and Daley compiled numerous other researchers' information and combined and tweaked their ideas to come out with the results that they did. The study numerous fossils and sediments accounts for all different time periods as well as the change in fossil structure over time. This slide shows more in-depth details about the study. The study focused on soft sediment environments because they are the most widespread and readily available. All samples contained at least 100 specimens and were from either North America or Europe. Overall results. The infaunal burrows increased about 30%. The bivalves increased about 10%. The predators also increased about 10%. The surficial dwellers increased about 40%. The completely non-motile animals, like the brachiopods, decreased about 30%. There is a possibility of preservational bias that changes these results. In both ancient times and current times, only 1 to 2% of animals were categorized as pelagic. This could potentially be because they do not fossilize easily, or if they do fossilize, it is nearly impossible to find their fossils, possibly because they are so deep. Pictured is a pelagic animal that exists now, the fangtooth. Escargots through time, an energetic comparison of marine gastropod assemblies before and after the Mesozoic Marine Revolution. Before discussing this paper, this graph is the basic interpretation of diversity before, during, and after the Mesozoic era. The largest peak in quantity of genera occurs during the Cretaceous era, leading into the Cenozoic era. This peak was caused by the Mesozoic Marine Revolution, which encompasses the Triassic, Jurassic, and Cretaceous eras. This era was lasted from 252 to 66 million years ago. According to the chart, the highest extinction percentage occurred at the end of the Paleozoic era, which, in turn, was the start of the Mesozoic era. After rebuilding some diversity, the Permian-Triassic extinction occurred. However, mollusks reproduced and expanded, which led to other types of marine life to appear, namely corals, marine reptiles, and fish. The quick expansion led to the Mesozoic Marine Revolution. This revolution changed marine life for millions of years to come. According to research Seth Finnegan, Craig M. McLean, Matthew A. Kosnick, and Jonathan L. Payne, studies show that while comparing metabolic rate and body size of creatures before, during, and after the Mesozoic Marine Revolution, energy consumption increased dramatically. The study showed that marine life metabolisms increased consumption by about 150%. Although gastropods were predominantly studied in this research, it is believed that other types of marine life experienced the same change. The study was based on gastropods of the time, namely escargots. In order to find this information from the past, researchers used basic scaling principles in order to find the information about gastropods. In addition to scaling, the researchers used extant data to understand more about the gastropods of the past. During the study, there were a few possible sources. The first is environmental heterogeneity. This would mean that different sizes of gastropods could occur in different environments, such as smaller forms of escargots in a higher latitude. The second is a lithification bias, meaning that it is harder to study or identify smaller types of gastropods because of the loose sediments compacting. The uncertainty is assigning metabolic rates is also the factor by excluding the fact that larger gastropods can have higher rate than the resting rate due to their predatorial habits. The temperature variability is also a factor because in more tropical environment, the deeper water is the same temperature as the shallow water in cooler places, which means the size may be affected at each depth because of the temperature. During this era, there was a definitive increase in energy budgets for gastropods. According to the evidence, gastropods included 8 times more energy from before the Mesozoic Marine Revolution to after. In addition to just gastropods, other species were likely to follow this trend. Occupation in Cenozoic sea mollusks. In order to better understand occupancy, we can take a look at ourselves as a species. We are well aware that our population continues to grow every day. In the graph above, it becomes clear that Homo sapiens have slowly but surely increased their occupancy and seem to be on an incline. A series occupancy is a combination of their ecological and biogeographical footprint. Every species' occupancy is different and varies with time. in order to better understand not only our species, but the species around us as well. It is important to recognize patterns found in nature. By doing so, and following the occupancies of the Cenozoic sea mollusks, we are able to derive a pattern and learn more about our future as a species, and the future of many others as well as understanding the past. The majority of this data set comes from 3 paleontologists who worked together, making their data extremely reliable, as there were 2 people checking each other's work at all times to avoid possible mistakes. Taxonomic consistency was very high. In order to make sure that this was the most accurate data set possible, data that was not extremely reliable was cut out. Data of mollusks who reside in the northernmost faunas of New Zealand experience extremely difficult weather patterns than the rest of these sea mollusks. This data was cut out because the weather could have been an adverse effect on occupancy. Data that lacked specific dates and was also erased from the data set because it was not helpful when attempting to derive a pattern. After cleansing the data, we are left with this final data set. This species was chosen because they wanted only to analyze species that occurred at least once in every stage with the strategic range in order to get more accurate and specific pattern. There were no sparsely occurring species so that trends could be more easily recognizable. Species as well as genera were tested in order to ensure that occupancy patterns were not species-specific, and more general that could be related to any type of genera or species. The method used with this data is simply counting the amount within a species found at given times. The final data was compromised on 2007 from 6,885 to 3,974 collections. 140 extinct well-sampled species, a span over 17 stages, and occurred over 40 million years. The results. The most common tendency is 56% of the data. Occupancy increases at first appearance. Short-lived maximum in the middle, and decline permanently after maximum has been released. In the figure on the left, it represents the graphs of a majority of the species that were tested. The ones that are filled in black are the ones that are consistent with the norm. As you can tell, that the highest point is found in each of the middle, followed by a decline. The figure on the left represents an overall compromised average pattern of each of the data points, which represents a clear incline, maximum, and then decline. Although we derived a pattern, the answer as to why this pattern exists is still up in the air. Perhaps factors such as dispersal and competitive ability enable species to rapidly fill the ecological and geographical space available to them at any moment. These geographical spaces are then possibly controlled by slowly varying geological processes, such as the marine transgression and regression. Species show no tendency to remain at their maximum occupancy once it is established. Species that have already been on a steady decline have the greatest risk for extinction. Species' presence over a greater geographic range helps to confer extinction resistance. So why? Although we derived a pattern, the answer as to why this pattern exists is still unknown. Possible factors such as dispersal and competitive ability enable species to rapidly fill the ecological and geographic space that becomes available at any time. These geographical spaces are then possibly controlled by slowly varying geological processes, including marine transgression and regression. Using species that have been around for an extremely long time is necessary because they have hints of both the past and future and can help us understand ourselves and the species around us.